like a summary reaction. We said you have food going in the form of sugar, so C6H12O6 plus oxygen. You are inhaling oxygen, but 6 molecules of oxygen going in. So keep in mind, food is going in, that's the sugar, and oxygen is going in, that's the oxygen we are inhaling or breathing in. We end up breathing out 6 CO2, the CO2, plus we form urine, and mostly we have water in urine, and we sweat, plus energy in the form of adenosine triphosphate. We know quite a bit more than this already. So what else do we know? We know that cellular respiration is a cascade of redox events. OX for oxidation and the red for reduction, and we defined those in the past. And before I break them down again, remember the way to remember it, as I said, sugar is going in from food, oxygen is going in, what you are, I said, breathing in or inhaling, and the CO2 is a waste. That's something you are releasing to the outside environment, and the H2O is a waste. This is what you have mainly in urine and in the sweat. So this is actually called all right, and energy in the form of cellular energy, which is adenosine triphosphate. Now the term redox one more time. So I will remind you. So going back, let's erase this. We said oxidation is losing the hydrogen, and the entire reaction is sugar is losing the hydrogen to the oxygen. So the sugar is losing hydrogen and becoming CNO, which is the CO2 here. So that's losing the hydrogen to become CO2. That's oxidation. And the O2 is gaining the hydrogen and becoming reduced into HNO, which is this. So that's your reduction. All that is also in the textbook, but we did that before. I said here, the goal, clearly these are wastes. So that's the goal. The goal is cellular energy, or adenosine triphosphate energy. So it's an energy transformation reaction. You are using or you are taking in this form of energy, and you will come up with this form of energy. All right, now, still remind you of a few things we did in the past about the energy. Going back to the reaction, write it down one more time. Same reaction, but I want to do this. Your book, the textbook, is not like it has more, like I want to round up and round down to simplify it. When I say energy, we have to agree that you end up with, if you use a glucose, C6H12O6, you will see that we want to stick to this 36 or 38 adenosine triphosphate. So I'm decoding this adenosine triphosphate energy, like how much or how many molecules are being generated from one molecule of sugar. Now, that may not match exactly or precisely what the textbook is actually claiming. And you will see why, but I said round up, round down, as we will see, than to deal with the decimals and fractions. All right, I also said in the past that every sugar molecule has in it approximately 686 kilocalories. That's a number you have to memorize. And every adenosine triphosphate has about 7. So if this is your 100%, when we do the math, and we will do the math in a sec, that is about, I mean, I can round up and say about 40%. It's even less than that. But let me show you where the 40% is coming from. On a very good day, you will end up with 38 adenosine triphosphate. On a bad day, you will have 36. Let's look at the 38. Be very optimistic. So 38, and every one of them has 7 kilocalories, and divide by the number you started with, 686. That is the roughly 40%. It's not 40, it's definitely less than 40%, but we can live with 40. But remember, the first, like according to thermodynamics, first law of thermodynamics, you can't create energy, you can't lose energy, you can only transform one form of energy into another. So I guess I'm missing 60%, which is released in a different form of energy, 60% in the form of heat, which is, all right, now, all this has been done before. None of this is new. Next, we also know, again, I said I'm reminding you of things that we did in the past. I want to visit or revisit the mitochondrion. We know this organelle is membrane bound, which means it has a membrane, a fence membrane around it. And we also said that this organelle is present in eukaryotic cells. There is one major exception. An exception to that will be red blood cells, and these are erythrocytes. These guys lack the organelle mitochondrion. All right. Now, the organelle from before, I said it's membrane bound. It has 2 membranes, the outer membrane and the inner membrane. But I said that inner membrane is forming those finger-like projections. Please review the textbook as well. I mean, all the way around. So let's label this here. This is the inner membrane. And the space in between the 2 membranes right there, that's intermembrane space. And every one of these guys, let's see. Let's do this. This one here, that's a cresta. Which is 1. And crestae, we did use, I said, this language before. The space and the fluid inside is called mitochondrial matrix. Look it up too. See how any of, or basically any term that we come across, look it up. See how it exactly is being defined. To make sure you understand what exactly the mitochondrial matrix is, or what exactly a cresta is, and so forth. I want to say this is basically what we know. The reaction itself, which is a summary reaction, As we will see, this reaction is taking place in many reactions. But the goal of the reaction is to make, excuse me, cellular energy in the form of adenosine triphosphate. You should review the structure of adenosine triphosphate. You should remember that this is a nucleotide. And you should know the monomers of that specific nucleotide. Like I would just mention them, it has the sugar ribose, the nitrogenous base adenine, and 3 phosphate groups. So sugar going in, a form of energy, adenosine triphosphate coming out, a form of energy. And I said we are transforming one form of energy into another. We started with 100%. We must have 100% when we are done. All right. So everything I'm going to say now is new. Yes, you have, excuse me, notes in the drive. Beginning with this, cells, human cells, are classified into either one, obligate or strict aerobes. And from the name, they must use oxygen. Otherwise, you can't make it. So the best example I can think of is our brain cells. I'm sure we all heard of brain damage. And why will we have brain damage if our brain cells are deprived of oxygen for some time? So you end up suffering from some sort of brain damage. And once again, because the brain cells can't survive without oxygen. I want to say most of your cells, human cells, will fall into this category. Give me 1 second, guys. Reply to a student. All right, sorry about that. The other extreme is obligate or strict anaerobes. They are the exact opposite. These cells will never ever use oxygen. And guess what? Remember, they don't even have the mitochondria. So the best example, erythrocytes, red blood cells. Now look how neat that is. Red blood cells do what for living? They transport oxygen. That's what they are doing, carrying oxygen around. But they themselves don't use it. So this is ensuring, I guess, 100% efficiency in transporting. They are not using the fuel they are carrying. So once again, they are carrying oxygen. If you loaded them with this much oxygen at A, they will deliver same exact much at B, because I said they themselves are not tempted to use oxygen. They don't care for oxygen. They don't even know how to use it. They don't even have the organelle that will use it, which is the mitochondria. So these cells are the best example of obligate anaerobes or strict anaerobes. Now we have the facultative anaerobes. Now English, let's use English really. Anaerobes, I guess, you know, anaerobes, you're not using oxygen. Facultative in English, what does it mean? Like you can look it up. It's not really a biological term, it's English. You study facultatively. You know what that means? When you must. But you don't really care to study, but you will study when you need to. So I honestly could use it, could use it in that sentence. You don't eat meat. You hate meat. But guess what? You're starving, and the only food I'm offering you, I mean, you have to eat or you will starve. then I guess you will eat meat. But now you are eating that meat facultatively again. So you are a facultative carnivores, let's say. You typically don't eat meat, but if you are in the middle of nowhere and that's the only food available, so you will eat it. So break it down then. Then I'm not aerobic. I'm sorry, I'm not anaerobic typically, but I will be so if that's the only option available to me. That's really the exact English translation of the descriptive language. So I will repeat again. Anaerobes, I'm not using oxygen, but facultatively, meaning I will only manage to survive without oxygen if I must. So I'm not enjoying this punishment, but at the same time, I will manage. So let's say if I want to try to come up with a fancier definition or breaking down, I will say these cells that prefer oxygen, but yet they switch to fermentation, which is anaerobic respiration, to fermentation when oxygen is absent. So I said, if you want to punish them, then take oxygen away. Will they die? They will not die, but they are not exactly excited about it. So I will say here, but they switch to fermentation. Or if I want to, I could say oxygen is present. Look what I'm going to write down. These cells are happy. Oxygen is absent. Well, I'm going to say they somehow, or I should do it this way, somehow manage unhappily. Somehow manage unhappily, or somehow unhappily manage, whatever. I hope it makes sense. So the best example, human muscle cells. Now, what I need you to do, this is the first assignment with this video. We have, when I say human muscle cells, well, guess what? Human muscle cells are of 3 different types. I'm sure you heard of this. I'm sure you heard of this, cardiac, and I'm sure you heard of smooth muscle cells. When I say muscle cells, which muscle cells? I will tell you, yes, they qualify here. Now I want you to look up these cells. Think about it. Do you expect cardiac muscle cells to be facultative anaerobes? Meaning, if you deprive the cardiac muscle cells from oxygen, will they somehow manage unhappily or unhappily manage? Will they switch to fermentation? So look this up. Smooth muscle cells, like cells in the lining, let's say the GI tract, like you have smooth muscle cells in the esophagus. So look into those as well. So this is the assignment. Look up those muscles and look up those muscles, please. Especially, this is, I see it's a huge deal. Like, hey, what's up with the cardiac muscle cells? But I said these cells, no question, that applies to them. What applies to them? That they will use oxygen happily, and when you take oxygen away, they will be upset, disgusted with life, but they still will lead a very unpleasant life, but they still lead some sort of life. Unlike brain cells. Brain cells, you have oxygen, they are having fun. No oxygen, they are dead. So big, huge difference. All right. Cellular respiration. We will break it down into 4 cascades. 1, or I guess A, glycolysis. And glycolysis is in the cytoplasm. Glycolysis in the cytoplasm, which means not inside the mitochondrion. And it's also another thing, oxygen independent. I'm using that language because a lot of books will describe glycolysis as being anaerobic. I have an issue with that term. When you say glycolysis anaerobic, to me, that implies glycolysis requires the absence of oxygen. To me, that's what that means. So I think they are being very loose in that language. So the glycolysis, I said, maybe your book is doing the same thing. I will write it down and scratch it out. I said, some books, including your book, perhaps, I really don't know, might describe it as being anaerobic. I said, I have a problem with that descriptive language, because when you describe something as being anaerobic, I said, to me, that means the absence of oxygen is a requirement for glycolysis to proceed. That's not the case. It's oxygen independent. It doesn't care about oxygen. Whether oxygen is there or not, I will not even notice. That's how much I don't care about oxygen. When I describe, let's say, Clostridium, that's a bacterium. When I say Clostridium is anaerobic, and actually it is, that means it doesn't use oxygen. And guess what? It doesn't use oxygen. Meaning what again? Meaning if you expose Clostridium to oxygen, you will kill them. They will not grow. All right, anyways. So enough about glycolysis for now. Not like enough, we're done with it, but it's different than the other three phases, B, C, and D. B, I'm going to call it for now transition phase. Of course, we'll come up with more names for it once we know what the heck that is. Krebs cycle, we'll see what that is, and also we'll come up with more names for it. And D, the last one, electron transport system. Those require oxygen. That's one. They are inside the mitochondrion. They have to have a mitochondrion. So they are very different than the very first phase. And now when I say inside the mitochondrion, where exactly? B and C, I'm referring to the transition and the Krebs. Where are they? In the mitochondrial matrix. Whereas D, this one here, this is location-wise will be cristae in the cristae. And we'll see, of course, where exactly. All right, so quick review. We said we are breaking down cellular respiration into glycolysis, which is in the cytoplasm, doesn't care about oxygen, doesn't care about the mitochondrion. B, C, and D, transition, Krebs, and electron transport, they require oxygen and they are inside the mitochondrion. Transition and Krebs precisely in the mitochondrial matrix, whereas D, which is electron transport system in the cristae, the finger-like projections. Back to our diagram here. All right. Glycolysis. What do I know? I'm going to still repeat. In the cytoplasm, old news, oxygen independent, which means will take place, I'm repeating again, with oxygen, or I should say in the presence of oxygen and in the absence of oxygen. It is a 10 cascade reaction or process. So it does consist of 10 reactions. Phases of glycolysis. Phase 1. Consists of 3 reactions. I will come back and come up with a name for that phase. Phase 2. Guys, I mean, I don't know how the book is breaking glycolysis down. I honestly have no clue. I can break down whatever any way I wish to break them down, as long as it's justified. So if the book is breaking them down into, let's say, 2 phases, and I'm breaking down into 17 phases, I will tell you why I'm doing so. And the book, of course, will tell you why they are doing so. So everything will be justified. So phases of glycolysis again. Phase 1, I said I'm going to break it down into 3 phases. Phase 2 consists of 2 reactions. Phase, at least according to my classification scheme, this one consists of 1 reaction. Now, what did I see here? Nothing. Phase 4 consists of, let's see here, 4 reactions. That's it. I guess I can't add anymore. Why? Because I have 3, 5, 6, 7, 8, 9, 10. And remember, I said it is a 10 cascade reaction. So once I add those up, that will be, they will add up to my 10 reactions. All right, phase 1. Phase 1. I know, once again, it's 3 reactions. Let's look at the reactions first and then come up with a name for the phase. What you do is, and this is in the cytoplasm because glycolysis is in the cytoplasm, you use a glucose. There is an enzyme, of course. I will write it down in a second. Enough room, I guess, to write the enzyme. The name of the enzyme that will catalyze the first reaction is hexokinase. What you will do to the glucose, you will add a phosphate group to it. So you grab an adenosine triphosphate, you dephosphorylate the adenosine triphosphate. So you are taking a phosphate from the adenosine triphosphate. Then adenosine triphosphate is losing a phosphate and becoming adenosine diphosphate instead of triphosphate. And that phosphate is being added to the glucose. Where exactly? At the 6' position. So glucose 6-phosphate. This is the first reaction. So what did I do to the glucose? I phosphorylated the glucose. If I wanna describe this sugar, just because I have nothing better to do, what exactly I'm looking at right now? I'm looking at a monophosphorylated Remember, glucose is an aldehyde, but it's a hexo. So a monophosphorylated aldohydride. hexose, just to show you. I mean, I can use this language. No one is going to yell at me. No one is going to say, what the heck are you doing? Why not? It is. Can anyone say glucose 6-phosphate is not a monophosphorylated aldohexose? No one can say it is not. So it is, glucose is an aldohexose. It's a hexose, but it's an aldehyde. Monophosphorylated, it has one phosphate. And where is that phosphate? At the 6' position. By the way, I'm not going to sketch every single sugar, but if you, all what I'm saying to you, remember that glucose, I really do not want to even waste the time. Look at the picture, look at the diagrams in the textbook. You're just adding a phosphate group to the 6' position. I just want to do maybe this here. So let's say this is a glucose. I'm not going to show you anything other than the 6'. So which is this? CH2OH. So remember, 1', 2', 3', 4', 5', and this is a 6', the branch. So all what I'm doing is adding a phosphate to that 6' position. So here, remember 6', CH2OH, I just want to write them down on the other side this way here. CH2OH. So now the phosphate group, let me use red. Okay. That's a phosphate group. The typical dehydration synthesis we used to do, and that's how we did it before. Why this is any different? You take the hydrogen from the OH and the OH from the phosphate. So I'm going to make them go away. You don't see them anymore. And now I have what, green, let's say. Now I have this linkage, which should be a phosphoester linkage. Phosphoester linkage. So this is your glucose 6-phosphate. Of course, imagine if I want to sketch every single one, that will take forever. I'm not. But I don't want you to get intimidated by the names. The names are exceptionally descriptive, exceedingly descriptive of the structures. All right, so I will repeat again. All what we did in the first reaction is we phosphorylated glucose. But to phosphorylate glucose, we needed to dephosphorylate adenosine triphosphate. And that's why when we dephosphorylated adenosine triphosphate, it became adenosine diphosphate. And when we phosphorylated glucose, we added that phosphate, not anywhere, not any random place, we added it to the 6' position. Hence the name, glucose 6-phosphate. But notice, I just lost 7 kilocalories. I said before, every adenosine triphosphate, where did I write it down? At the very beginning, here. Earlier or below, right there. Every one of them. So I'm actually not at the zero, not like I did not make any money. No, I actually did lose 7. At least if I want to bail out right now, this is not very promising. It gets worse. All right, so the second reaction is I'm going to have glucose. So this is what I have right now. So glucose 6-phosphate, another enzyme. You transform glucose 6-phosphate into fructose 6-phosphate. I will write the name of the enzyme in a second. But what's the difference? Not much, except now you have a ketone instead of an aldehyde. So I'm not going to sketch it, guys, because that is wasting a lot of time. All what I'm saying is there is an enzyme sitting here that will transform the glucose 6-phosphate into a fructose 6-phosphate, which is your second reaction in the first phase of glycolysis. The name of that enzyme is phosphoglucoisomerase. Look, they are in the book. They are online. Look him up. Phosphoglucoisomerase. Look, it has isomerase in it because it's creating an isomer, because fructose and glucose are structural isomers. Phosphoglucoisomerase. All right, then you grab the fructose 6-phosphate, and guess what? You actually phosphorylate this yet again. So you are using another adenosine triphosphate. So you will dephosphorylate that. So adenosine triphosphate will become adenosine diphosphate. And you add that phosphate to the fructose 6-phosphate at the 1' position. That's why you end up with fructose 1,6-bis phosphate. You can say bisphosphate, diphosphate, I don't care. But it is exactly that. So if I want to go ahead and sketch it, I'm going to have a fructose. I'm going to have a phosphate at the 1' position and a phosphate at the 6' position. So if I want to exercise my naming business, I will say now I have a diphosphorylated ketohexose. Do I not? Fructose is a ketone, it's a hexose, and it has 2 phosphate group, fructose 1,6-bisphosphate. All right, and the name of the enzyme, phosphofructokinase. All right, going back. What did I say, phase 1? I said it does consist of 3 reactions. So I'm kind of done with phase 1. But if that's the case, I'm not exactly sure if I should feel accomplished, because I did lose 7 kilocalories here, right there. And I did lose 7 more here. Where are we going? Well, like business, you have to invest money to make money. But what's, without getting into a lot of chemistry either, but think of it that way here. What I'm trying to do is, this is my own invention. You're not going to find this anywhere. I'm trying to convince you that it is wise to do so. Let's say glucose is sitting here. Let's say so here, G for glucose. This is time, and this is the energy level here. E, which is energy, in kilocalories, let's say so. So that's where the glucose is. What do I do is, of course, I said this is time. Over time, I will end up with this dude here, which is glucose, forget the dashes, glucose 6-phosphate. Notice it is at a higher energy level. Of course it should be. Let me use red. Because what did I do to move from here to there like this? What did I do? Did I not do this? Let's use yellow. I did this. I used an adenosine triphosphate, and I took that phosphate from adenosine triphosphate and I transferred that to the glucose. That's a phosphate. And the glucose became a glucose, like the glucose here became glucose 6-phosphate, and the adenosine triphosphate became adenosine diphosphate. Bear with me. So what I'm trying to do is excite the sugar. Excite the sugar to do what? Still bear with me. Then over time, once again, I ended up with this, which is fructose 6-phosphate. But notice, I did not have it at a higher energy level, because I didn't add any more phosphate to it. Then I had this one here, which was fructose 1,6-bis. Can I do it this way? Bisphosphate. Here. Fructose 1,6-bisphosphate. Same thing. Let me use it still yellow. I did this to get from here to here. Same exact thing. I use an adenosine triphosphate. I took the phosphate from it. And added it to the fructose 6-phosphate. That's why it's now fructose 1,6-bisphosphate. And adenosine triphosphate is now adenosine diphosphate. So I started, let's use black. I started here at this energy level right there. And this is where I am right now. This is, let's say, my new energy level. Right here. Why so? Because guess what? I said it's the second I get to this level, the reaction, look what I'm going to do, will proceed spontaneously. The reaction will not proceed spontaneously if I have only this much energy or only this much energy. I have to have that much energy for the reaction to proceed spontaneously. We are not going to get into transition phase and none of that, or the energy of activation. All what I'm saying, think of it that way. Just, you know what? You are making, let me use this silly analogy. See, you need the sugar to jump, because when the sugar jumps like this, that's what I mean by jump, you start making money. But how will you make the sugar jumps? You have to make the sugar somewhat less inhibited to jump. Excite the sugar, make the sugar drunk by adding phosphate to it. So this is where the sugar was, right there again. The sugar was very unmotivated to do much, not acting silly or crazy. Getting there. Well, I made it happen. The sugar right now is very less inhibited. I said let's really use the term drunk sugar. Then the sugar will jump. And the second it's jumping, I'm going to start making money. This is where money is made. So I'm going to go back then to the first phase. What did I call it? Nothing yet. I just said phase 1 consists of 3 phases. No one will argue with me if I called it here phase 1, if I called it sugar, or to be more exact, sugar's activation, or even sugar's phosphorylation. Because all what I'm saying to you is, did we not activate the sugar? We did, right? Because that's the active sugar. And how did we go about it? By phosphorylating the sugar. So sugar's activation or sugar's phosphorylation. Now, when I say sugars, why sugars? Why S? Because which sugars did we activate, or which sugars did we phosphorylate? Did we not phosphorylate glucose and fructose 6-phosphate? These are the 2 sugars that have been phosphorylated. Those 2, glucose and fructose 6-phosphate, they have been phosphorylated right there. In the first reaction, the glucose, look, is being phosphorylated. And not the second reaction, but the third reaction. Oops, is it the one? Yes. Here, fructose 6-phosphate is being phosphorylated. So all what we are doing in this phase is adding phosphate to activate, to make the reaction spontaneous, to make the reaction happen. This is the kickstart. Think of it that way. you need here, you need roughly 14 adenosine, sorry, 14, not what I meant, 14 kilocalories. Why 14? Because we use 7 and 7, 2 ATP. 14 kilocalories here, 2 adenosine triphosphate, to kick start the reaction. So that's, some books, by the way, will call that phase the investment phase. Okay, whatever. Call it whatever you want to call it. I said if it's making sense. Here, the investment phase. If makes you feel good. Why investment? Because adenosine triphosphate has been invested. But I also justified my logic, my philosophical twist. I said sugars activation or sugars phosphorylation. All right, 3 reactions, and so far I'm losing money. Next phase, look, I said 2 reactions. Where are we again? Here. It's 2 reactions. So what will happen? What do I have right now? Like, let's do inventory. There is not much to do. I just have one molecule of fructose, 1,6-bisphosphate. One molecule of fructose, 1,6-bisphosphate. Guys, this is the one I'm talking about. Here. It's this, but I want to go back to this reaction right there. This is the one, fructose 1,6-bisphosphate. Forget ketone aldehyde. Doesn't it look like this? It's a 6 carbon sugar. So 1, 2, 3, 4, 5, 6. Let's say the open form. And then you have the phosphate. Let's use red for it. Phosphate group. 1' or 6' phosphate group on the other end. That's basically, essentially what it is. You do 2 things because it's 2 reactions. Once again, 2 reactions. You split. So when you split, you end up with one sugar that will look like this. I will add the phosphate, and I will add the phosphate here. Let's finish this, red. When you initially split the 2 trioses we mentioned in this class, because now what do you have? You have 8 triose, right? 1, 2, 3, another triose. But every one of them has a phosphate group. So you end up with a monophosphorylated triose and a monophosphorylated triose. One of them is dihydroxyacetone, dihydroxyacetone phosphate, which is one of the, this is a trisaccharide that we mentioned before. Phosphate because it has a phosphate group. And the other one is actually glyceraldehyde phosphate. Glyceraldehyde phosphate. That's the other triose. Go back and review the 2 sugars, the 2 trioses we mentioned. One of them is dihydroxyacetone, the other one is glyceraldehyde. All right, but that's looks like one reaction. I'm gonna say here, I'm gonna write the enzyme, which is enzyme, the 4th enzyme. But before I do, I have an issue with this name. Which name? This one right there. Why? What's wrong with glyceraldehyde phosphate? I'm perfectly okay with dihydroxyacetone phosphate, but I'm not exactly okay with glyceraldehyde phosphate. I wish again we were face to face so we can ask these questions and wait for you to tell me why, because it's a very nice recap or review. Dihydroxyacetone, forget the phosphate. Dihydroxyacetone is this. It's a ketone. And I don't care about orientation right now. This is dihydroxyacetone. Bear with me, not dihydroxyacetone phosphate, just dihydroxyacetone. Now, glyceraldehyde was this. It's an aldehyde. I don't know how much you remember from that business. All right, where I'm going with this. When I say dihydroxyacetone phosphate, where are you adding the phosphate? Well, there is only one place to add the phosphate, here, because this one is the same. That's not a different location, it's the same. But when you look at the glyceraldehyde, this location is not the same as this location. These are two different locations. Because this molecule is very symmetric, but this is not symmetric. This is asymmetric. So in the case of dihydroxyacetone phosphate, I know where the phosphate group is. The phosphate group is either here or there, but that's the same location. That's not two different locations. So that's why for glyceraldehyde phosphate, I have to specify where is it. It's actually glyceraldehyde 3 phosphate. It's added here. That's where the phosphate is added. So if I want to go back and add it, and I will, that's fine. Sorry. Let's erase this. All right. It is 1, 2, 3. Now OH, right? OH. So remember what we do. We take the hydrogen from the OH. So I still have the O. But the hydrogen is gone. I said we take the hydrogen from the OH. Now a phosphate group. Let's do it in black. Sorry. The O. The OH. But I'm taking that. Let's actually not. Let's fix it. All right. The phosphate group here. So I'm going to take the OH like this, and now I have this linkage, which is an R-phosphoester linkage. But now if this phosphate was here, that's a different molecule. All right, so reaction number 4, and I said I will write the enzyme in a sec. You end up with dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. I'm gonna